Okay, hi everyone. Welcome to another video. This video is about DNA structure and also DNA replication. So there's two pages here on OneNote that I have written on. They're visible in my notes section if you want to look at them. I also put in my notes section the actual slides for this, but I'm just going to sort of talk through my notes that I have. Oh no, there we go. What's happening? It's mad at me. Alrighty. So first of all, hopefully you just watched a video about the discovery of DNA, or if you haven't done it in that order, you're going to watch it in just a moment after you watch this. But DNA was discovered by two men, Watson and Crick, in the 1950s. So really not that long ago that DNA was discovered. There was a woman, Rosalind Franklin, who this was in the video, but I'm going to summarize it for you because I'm passionate about it. Who took this photo using X-ray crystallography to show that DNA actually had a double helix shape. Because before that, Watson and Crick were like kind of thinking about a triple helix situation of DNA, which obviously is wrong. And Watson and Crick and other scientists also thought that the bases of DNA must point outwards because that's where the information is. So for the information to be read, it needed to be on the outside. This picture by Rosalind Franklin like really changed the way that people thought about DNA because it cemented that it was a double helix because of the X shape and not a triple helix. The issue was and is that Rosalind Franklin did not like give permission for people to have this photo. Basically her assistant like went into her desk and took the photo and gave it to Watson and Crick, and they were like, here. Which is obviously not okay. And then also Rosalind Franklin doesn't get credit for like the discovery of DNA the way that she should, partially because she died before she could be awarded a Nobel Prize, which is not awarded after death, and also just because she was a woman. So remember Roz when you think about DNA because she was really important to discovering it. There are a few other like players in this game. Hershey Chase was an experiment that used bacteriophages, which are viruses, to show that the viral DNA is what entered the cells and caused like changes in the cell to make them into virus producing factories. But bacteriophages are viruses that infect bacteria. A group of scientists used pathogenic bacteria to show that DNA could change organisms. Because for a while, people really were not sure whether it was DNA or proteins that was really the genetic material that was like coding for traits. So these experiments helped people to realize, oh, it is DNA. That's what matters. There's Wilkins. He's the one who actually like gave the photo to Watson and Crick. So curse you, Wilkins. Up at the top is just some general information about DNA. So it is a little bit different in prokaryotes versus eukaryotes. In prokaryotes, there are circular chromosomes. And in eukaryotes, like us, we have linear chromosomes or X-shaped if it's after DNA replication. And then there are also plasmids, which are small circular units of DNA that are found in mostly bacteria, but also can be found in viruses and eukaryotes as well. And they're really small units of DNA that have only a few genes. They're really important for bacteria to pass like antibiotic resistance and other traits to one another using plasmids. I'm going to talk about them more when we talk about biotechnology. But down here, I drew a little picture of a prokaryote, a bacteria. The chromosome is large, but if you unraveled it, it would be circular. And then the plasmid is like a super tiny little donut of DNA right there on the side. And they could have multiple plasmids. They can pick up plasmids from the environment. They can exchange them, but they're still DNA. They're just circular units of it. All right, looking at the structure of DNA, this is probably something that's familiar to you from previous biology classes. Here's the double helix in like a cartoony version, and then the actual protein and nucleic acid structure is over to the right side. the molecular model. And in the middle is sort of a zoom in of the hydrogen bonds and covalent bonds and different interactions that are happening within the helix. A couple important things about this. So the blue lines that are the outside of the DNA structure, they're called the backbone. They're made up of sugars and phosphates that are covalently bonded to one another. And then inside of that, the sort of rungs of the ladder are the actual nitrogenous bases, which remember from previous classes are A's, T's, C's, and G's. A always binds with T and C always binds with G, which I usually tell people that go here to remember at Cardinal Gibbons how they go together. The different bases have different names. So A and G are called purines, and T and C are called pyrimidines. The purines are larger than the pyrimidines. They have two rings, two carbon rings, instead of having just one. You can see that here in the picture. The A is a purine and it's larger, and T is a pyrimidine and it's smaller. And it's important that a purine goes with a pyrimidine because that keeps the width of the rungs the same. Because if it was two small ones matched to two big ones matched to two small ones, the DNA ladder would be kind of wavy and wonky. And it isn't like that. It's the same width from backbone to backbone. And that's because of a purine and a pyrimidine matching together. The other thing that's important here is that DNA runs in two different directions. So there are two strands. The middle of the strand is hydrogen bonded together. It allows it to be unzipped during replication. The fact that they run in two different directions is called anti-parallel. Here it is, which means basically one is right side up and the other is upside down. In DNA, instead of saying like right side up and upside down, we use numbers as a reference of the direction for which strand is pointed up and which strand is pointed down. And what we use for that is 5 prime. So this apostrophe is pronounced as prime, and 3 prime. So the 5 prime carbon, this refers to the carbons that are on deoxyribose, which is the sugar that makes up DNA. It's the blue pentagons in the picture. So on the 5 prime, that carbon is attached to the phosphate group, which you can see is the yellow circle in the picture. On the 3 prime end, the carbon is attached to a hydroxyl group, which is that OH. That's how you tell the difference between the ends. 5 prime is attached to phosphate and 3 prime is attached to hydroxyl. If we break down these monomers all the way, they're called nucleotides. I drew in this little picture a base that's just a rectangle. But obviously they are either purines or pyrimidines, so they're a little bit different depending on what they are. The sugar, though, is always deoxyribose. It's a 5 carbon sugar. And then, of course, that 5 prime carbon is attached to the phosphate group. And that is the structure of DNA. So to put it all together, it's a double helix. It's anti-parallel, so you have strands running in opposite directions. 5 prime is attached to the phosphate group. 3 prime is attached to the hydroxyl group. And you have different types of bases. A always goes with T. C always goes with G. A and G are purines, and T and C are pyrimidines. The purines are larger than the pyrimidines. Okay. Now let's talk about how DNA replicates. This page is also in my OneNote. First of all, DNA replication happens during S phase, which is part of interphase. It's the part of interphase where the DNA replicates. S stands for synthesis. One thing that's important about DNA replication is that it always happens from 3 prime to 5 prime on the template strand. So what that means is that because DNA is anti-parallel, the two strands have to do this a little bit differently. So when replication happens, the strands are unzipped down the middle. There's an enzyme that does that called helicase. Here it is. It's the green blob. It's moving down and unzipping the strands from one another. There are some single strand binding proteins. They're gray blobs in this picture. The color of them is irrelevant, but they are for this one. Their job is to make sure that the single strands remain stable and also are able to be replicated. Your cells are good at recognizing single strands of DNA as errors and mutations and break them down. So single strand binding proteins sort of stabilize that and make sure that it doesn't happen. Also, there's an enzyme called primase. Primase's job is to add RNA primers onto the empty strands. The primers have to be there in order for replication to start. And they'll be removed after, but they're really important for actually starting replication. And then there's one other blob over here, topoisomerase. Its job is related to the twisting and untwisting of the helix, which you'll be able to see in other pictures too. But helicase, topoisomerase, and primase in this picture are all enzymes. They're proteins that are responsible for helping with replication. All right, so back to the two strands. The strand that runs from 3' to 5' can be copied continuously because that's the way DNA replication works. It just only works in one direction. So that strand is called the leading strand because it can go a lot faster than the other one. The other strand is called the lagging strand. So it's running from 5' to 3'. It has to be copied in little fragments backwards. And those fragments have a special name. They're called Okazaki fragments. And they're later bound together by ligase, which is another enzyme that is related to this. Before I go any further, I just want to show the organization of DNA in your cells. So here's this diagram. It's really large. And it's really large because you have so much DNA and it's hard to organize it. So first of all, you have a double helix. That's your DNA. It is only 2 nanometers in diameter. So it's super, super, super small. Your cell would have a really hard time keeping this organized if it didn't have a neat and clean way to do it, which is called a chromosome. So in order to create chromosomes, your DNA coils around histones, which you can think about them as sort of like a spool, where the DNA is the thread. The histones come together to create a complex of 8 histones, and then just like a thread on a spool, DNA wraps around the histones. And then the histones coil up together. So you have this small coil, and then you have a bigger coil of DNA happening. The bigger coil, which now we're up to about 30 nanometers in thickness, is called chromatin. And chromatin is still pretty disorganized in cells. You can't see the individual strings of chromatin underneath a microscope. These images are a really intense electron microscope. So then chromatin forms into loops and eventually coils up into a chromosome, which is a unit of DNA that you actually can see under a microscope. And you have 46 of these chromosomes in every single one of your cells. So that's a ton of DNA. Each of your chromosomes has millions of base pairs, that's A's, T's, C's, and G's, that your cell has to keep track of and make sure that it all gets replicated in order for your cell to do mitosis and copy. Also, I made the note that the X-shaped chromosome is replicated, so that would be after S phase. Before S phase, the chromosome is just linear, so it would just be a regular line like this. After replication, you get the exact copy of the other half like this, but both the left and the right would be identical to each other because they would have been copied from one another. So that's the organization of DNA. Hopefully it feels amazing to you, because it is. All right, here's the enzyme list. I already talked about these a little bit, but I just made a handy little list for you. Ligase binds together the Okazaki fragments only on the lagging strand. DNA polymerase, I didn't talk about yet, but its job is to add new DNA nucleotides to the growing strand. So it's inserting the think about them as Lego pieces. DNA polymerase is inserting them. There is also RNA polymerase that we're going to talk about during transcription at the end of the week, but DNA polymerase creates a new polymer of DNA. Helicase is the zipper. Think about it like helix, because that's what it's related to. It unzips the hydrogen bonds in between the bases. Topoisomerase's job is to like swivel, break, and rejoin the strands throughout the replication process. It's the blob that was towards the far end of the previous picture. And then primase is responsible for sort of initiating DNA replication by adding the RNA primer. If we look at this picture here, you can see that the replication process is happening in both directions. So there is a point right in the middle that's called the origin of replication, because the replications are going in opposite directions from the origin. They're going to work differently on either side of the origin. You can see on the left side of the origin, the top strand is leading and the bottom strand is lagging. It's the opposite going the other way. And that's because you're basically working backwards on that side. The little red sections are meant to show the primers. And you can see the Okazaki fragments on the lagging strand that then would have to be bound together by by ligase at the end. And you can also see in this picture down here that this DNA polymerase is DNA polymerase 3. There are a few different ones. They all are responsible for adding DNA nucleotides to the growing strand. And then the origin of replication is just the starting point of replication. Remember, prime to 5' is going to be the leading strand, and 5' to 3' on the parental DNA or the original DNA is going to be the lagging strand. All right, so just a little bit more about this, almost done. DNA replication is a semi-conservative process, and what that means is that each new DNA strand will contain half of the old DNA strand. So in this image, I took my blue double helix, split it apart, and then the red side is added in. So each time DNA replicates, half of the old one is now contained in the new one because it was used as a template to create it. Just a little differences between bacteria and us. In bacteria, replication has one point of origin, which means it begins at only one place on the chromosome. For us, there can be multiple origins of replication, which means that your DNA can replicate beginning at several different points on your chromosomes. So it allows it to happen a lot faster because you can start in many different places at the same time. Finally, DNA replication will end with a proofreading step to check for mutations before proceeding into mitosis. If the cell finds mutations, it will do one of three things. One of them is fix the mutations, because your cells are actually really good at that. The second one is exit the cell cycle and never divide again. That's called G0. And the third option is to perform apoptosis, which is programmed cell death. So your cells actually will destroy themselves for the good of the other cells, because if mutations continue to build up in the replicated cells, it can lead to cancer. So replication is a really important process that your cells get right almost all the time, but it's happening super fast. So mutations are always possible in randomly occurring events. Okay, that's it, guys. Great job. There's a little matching thing for you to try on Google Classroom. So check that out, and then you can save that to your notes for future use. But great job. Keep up the good work, and see you soon.